Just wanted to talk about this 2015 study we had in JBC. It's a pretty cool investigation of what happens when you isolate deubiquitinases or DUBs from the proteasome and how they interact with the polyubiquitin conjugates that they'd normally find in the cell. For this study, we did quite a bit of in vitro work, built some substrates, and then asked the question, how do each of these DUBs and the whole proteasome interact with these different conjugates of polyubiquitin? And with that, let's go into the notebook LM summary of this work.
All right, we're here to conduct a technical audit of the Mansour et al. Paper. The focus is specifically on their characterization of two proteasomal deubiquitinases, Rpn11 and Ubp6.
Here's how we'll structure this audit. First, the research objective and design. Second, the polyubiquitin substrate engineering. Third, we'll dig into the comparative enzyme kinetics. Fourth, the mixed linkage substrate disassembly. And finally, we'll wrap up with a technical summary of the findings.
So let's start with the core objective. The goal here was to characterize the intrinsic enzymatic properties of these proteasomal DUBs. And the key was doing this in isolation, separating them from all the ATP-dependent processes you'd find in the intact proteasome.
All right, the two DUBs in question here are quite different beasts. You've got Rpn11, which is an integral zinc metalloprotease. It's part of the 19S lids architecture. Then there's Ubp6, a cysteine protease that just sort of transiently docks onto the proteasome via the Rpn1 subunit. This is a really fundamental distinction, and it's key to understanding their different activities.
So the whole experimental premise, really, is to pull these DUBs out of the full proteasome complex. They used a specific panel of non-degradable polyubiquitin conjugates to do this. The idea, of course, is to get rid of all those confounding variables, things like substrate translocation and degradation, which are normally all tied up with deubiquination in an ATP-dependent process.
Okay, moving on to section 2, the substrate engineering. This part is absolutely critical. I mean, the quality and the precise definition of these substrates, that's what underpins the validity of the entire kinetic analysis that follows.
Let's look at the substrate panel they put together. It was clearly built to test some very specific ideas. You've got these homogeneous polyubiquitin chains, Lys11, 48, and 63, which are perfect for probing linkage-specific processing. But then they also threw in something much more complex, an autoubiquitinated UBCH5B. This gives them a heterogeneously modified target, something a bit closer to what you might see in a real physiological context.
And here's the proof in the pudding, so to speak. This is the SDS page from their supplementary figure 3. It clearly shows the successful purification of that UBCH5B Ubin substrate. You can see how the hystrap purification did its job, getting rid of those contaminating free polyubiquitin chains. You know, this is a non-negotiable validation step before you can trust any of the downstream assays.
These models from subfigure S1 really get to the structural heart of the matter. On the left, you see the Lys48 linked dimer, and it's all bunched up in this compact globular shape. Compare that to the Lys63 on the right. It's much more open, much more extended, and it's these totally different topologies that the DUBs are thought to recognize. It's the structural basis for all the specificity we're about to look at.
All right, now we're getting to the real meat of the study. Section 3, the comparative enzyme kinetics of Rpn11 and Up6. This is where the quantitative data lives.
So to get to their numbers, they used this kinetic scheme you see here. It's a model for the disassembly of a tetraubiquitin chain. And it's clever because it accounts for two different types of cuts. Exocleavage, which is just snipping off the ubiquitin at the very end, and endocleavage, which is cutting somewhere in the middle of the chain. This lets them calculate the individual rate constants for each process.
Okay, let's look at the numbers. This table comes from subfigure 2, and it summarizes the key rate constants. The data point that just jumps out at you is UB6's activity on the K48 chains. Look at that. It has a strong preference for endocleavage. The rate constant is more than double that of exocleavage. And then you look at the isolated Rpn11 complex, and wow, it's just orders of magnitude less active, period.
So a really central observation comes out of all this kinetic data. When it comes to Lys48 linked chains, the longer the chain gets, the worse the processing efficiency. But for Lys11 and Lys63 linkages, it's the complete opposite. The enzymes actually get more efficient as the chains get longer. This points to a fundamentally different way the enzymes are interacting with these different ubiquitin chain structures.
Okay, so the next logical step for them was to use that heterogeneously modified UBCH5B UBN's substrate. This is where they can really test DUB specificity on a messy, more complex conjugates and get a better handle on Rpn11's role within the context of the full proteasome.
But of course, before you can test disassembly, you have to be absolutely sure what's in your complex substrate. So here we have an MS-MS spectrum from subfigure S4. And this is the hard molecular evidence showing that yes, there are non-canonical Lys11 linkages present in that UBCH5B Ubin conjugate.
And the data here just adds another layer to the substrate's complexity. Further, mass spec confirmed they also had Lys63 linkages in there, plus others. So once they validated all these different linkage types, they knew they had a suitable, complex substrate to really test for broad DUB specificity.
So what was the big takeaway from this assay? Well, the key finding is that the intact proteasome can just chew through this complex mixed linkage substrate. It disassembles it completely. And this activity seems to be coming primarily from the broad specificity of Rpn11. It looks like its activity gets a massive boost once it's incorporated into the full 26S complex.
Now, we do have to address a primary limitation of this study's in vitro design. Look, it's a great setup for isolating the intrinsic properties of the DUBs, but what it can't account for is that synergistic, ATP-dependent coupling of deubiquitization, substrate unfolding, and translocation. All the stuff that happens in the holoenzyme. And the proof is right there in the data. They note the intact proteasome is over 100 times more efficient.
All right, let's move to the conclusion, a technical summary of the findings and what they mean.
So, to summarize the technical findings, first, both of the isolated DUBs, Rpn11 and Ubp6, are actually better at processing Lys11 and Lys63 linkages than the classic Lys48 linkages. Second, Rpn11 itself shows a very broad specificity, which is what lets the proteasome handle those messy mixed linkage conjugates. Third, Rpn11's activity is massively potentiated when it's part of the full proteasome. And the final point, which is more of a proposal, is that the poor ability to disassemble long, homogeneous Lys48 chains might not be a bug, but a feature. It could be a way to prolong the substrate's residency time on the proteasome, maybe for some kind of proofreading step.
And this all leads to the study's final, pretty provocative statement. The fact that the canonical K48 chains are processed so inefficiently, well, that really calls into question the simple dogma of what a degradation signal is. It forces us to rethink how the proteasome interprets these different ubiquitin signals. You have to start distinguishing between a signal for just targeting and a signal for commitment to actual proteolysis. And that right there is a pretty significant conceptual shift for the whole ubiquitin field.